you're ready, you can go ahead.
Great. Well, let's get started. Thanks, everyone, for calling in. Full disclosure, I am not a true expert on transcripts. I'm just going to give you some background that I've picked up while doing gene and variant curation in the clinical lab, and I really do encourage you to check out the webinar recording that I'll refer to in this presentation when it is available, and the slides from NCBI and Ensembl EBI. So this is the outline of my talk today, and I should have enough time for questions, but feel free to interrupt me as we go if you have a question. So I'm going to start with why transcript curation is important, and I'm going to illustrate this by using examples from the hearing loss domain. Apologies, like Jenny said, I am largely doing a lot of hearing loss curation, so most of my examples, if not all of them, are hearing loss. And then I'm going to talk to you about transcript annotation bodies and the different transcript types of transcripts that you will see when you're doing your curation. Hopefully all of this is in the scope and in the context of doing curation. I tried to make it like that. And then I'll end with how can you curate transcripts, what are some of the resources that you can use, and then give you an example of actually how I did a systematic curation of basically 160 or 150 or so hearing loss genes that are on our clinical testing panel at the Lab for Molecular Medicine in Cambridge. So I definitely don't have to tell everyone on this call this because you're all biocurators, but I do like to remind people that variant interpretation requires gene curation. If a gene's not associated with disease, you really can't classify the clinical significance of a variant. And I think that this is illustrated best in the figure modified by Daniel MacArthur, where you have variant level evidence here and gene level evidence here. So if you have a very strong amount of gene level evidence and a very strong amount of variant level evidence, you have delta F, by the way, in CFTR, very strongly associated with cystic fibrosis. Maybe you don't have a ton of variant level evidence, but you are curating a variant in CFTR, which is definitively associated with cystic fibrosis. You end up with a VUS in a gene that's definitively associated with disease. If you don't know very much about the variant and you don't know very much about the gene, you end up with a VUS and a GUS. So a variant of uncertain significance and a gene of uncertain significance. You really can't go higher than this. And you can't have a variant with very high level evidence without high gene level evidence. Maybe the one exception is one of the first variants that you end up scoring in a gene curation. Maybe it's very convincing, it has a large family worth of segregation, but in general, you can't classify variants without classifying genes. And I hope in this talk to convince you that you really do need to do some legwork in transcript curation before you can classify genes or variants too. So variant interpretation may be dependent upon the transcript. We often want to assess the most severe molecular consequence. However, the transcript choice may also be dependent on the disease and the tissue-specific expression of the transcript. Maybe the variant could be represented as a missense variant or a nonsense variant, but your missense variant is actually expressed in the tissue of interest, and the nonsense variant is expressed in some random tissue that isn't associated with the disease. In general, clinical labs, because this transcript curation is time-consuming and there's often not a lot of information in the literature, will often choose the longest transcript for annotation. Just so they can be the most inclusive as possible. However, this might not be the most biologically relevant transcript, and we have many examples of that in hearing loss, actually. So I'm going to give you a couple examples now why transcript curation is important. And the first one is a tissue-specific exon expression. So this is the gene TBC1D24. It's associated with non-syndromic hearing loss, Doer's syndrome, which has nail and teeth and seizures and intellectual disability associated with it, and also a spectrum of epilepsy conditions. RefSeq, which I'll talk a little bit more about soon, lists 2 curated NM transcripts, and these are shown here. This one at the top is expressed in mouse neurons, and this one below is expressed in mouse cochlea and non-neuronal tissues. There happened to be a frameshift variant that was identified in the literature that was associated with severe lethal epileptic encephalopathy without hearing loss. because it causes a frame shift in this exon that is present in this transcript that is expressed only in mouse neurons and not in mouse cochlea. You could say that we don't have human expression data for this, and that's one of the challenges in the hearing loss field. But this is a pretty good example where transcript curation might be important. Another one, and I kind of alluded to this in the beginning, is an example of multiple molecular consequences. So you have the CLARIN1 gene, and it is associated with Usher syndrome, which is a syndrome associated with hearing and vision loss. RefSeq lists 4 transcripts, and they're all listed here, and also listed below as a screenshot from Element. The longest transcript is listed in green, and that's here, and the variant is a missense variant in that transcript. The reporting transcript that our lab uses that's largely present in ClinVar and the human gene mutation database is the one in blue. That's here, and that's also a missense variant on that transcript. However, there is also another transcript that's boxed in red where the variant is a nonsense variant. And in the case of Usher syndrome, it's tricky because reported likely path and path variants are actually a mixture of missense and nonsense variants. So in this case, it's a little bit difficult to decide which of these molecular consequences and transcripts you should choose for this curation. And functional studies don't really clarify because they show that the variant protein was mislocalized in the cell and rapidly degraded. So in this case, it turns out that the variant is pathogenic, and we did mention in our summary variant blurb when we submitted to ClinVar through the Lab for Molecular Medicine that we couldn't be sure which molecular consequence was causative for disease. But it is always important to consider the molecular consequence of each variant on each transcript and try to do some legwork for curation to decide which one to report or which one to analyze. This one did end up being likely pathogenic or pathogenic, I can't remember, so it didn't necessarily affect it, but it was important to note that there were multiple molecular consequences. So I want to talk to you now a little bit about transcript annotation efforts. And so 2 of the most common ones that you'll see are Ensembl and RefSeq. So the first one that I'll talk about is Ensembl. This is from Ensembl EBI. These transcripts are designated by automatic and manual annotation. I have worked a lot with the Ensembl EBI team and the NCBI team, and in the past it used to be the thought that most of this was largely automatic annotation and that they were just finding all the transcripts that could align to the genome build, and many of them didn't have evidence. However, they have assured me, and I'll show you a screenshot of the website, that they've been starting to do more and more manual annotation and manual curation of these transcripts. ENST is the label for Ensembl transcripts. You'll also see ENSP on the screenshot for the website, and that stands for protein. Most importantly, the references that use Ensembl transcripts are basically all of your population database references. So gnomAD, EXAC, 100,000 genomes. GTEx, which I'll talk about later, it's an expression database, Decipher, and also Cosmic. So that's important to keep in mind that clinical labs, and I'll get a little bit more into detail on this, often use RefSeq. However, these large population databases use Ensembl. So you often have to search using genomic coordinates to be sure that you're finding the right variant. And a big difference between Ensembl and RefSeq is that the sequence is predictive for Ensembl, and it's pulled directly from the genome build. So it's aligned directly to the most recent genome build. All of these references currently, gnomAD for sure uses GRCH 37, but hopefully everything will be migrating soon to 38. This is an example of a screenshot from Ensembl. I just searched the gene that we had talked about previously, TBC1D24. You can see all of the transcripts are listed here. Something that is really useful is actually they list in the table, and I'll talk about more about this when we come to curation, the corresponding RefSeq. It's really helpful to be able to map an Ensembl transcript to a RefSeq transcript if you need it, potentially if you're looking in GText to look at the expression levels of transcripts. It's really helpful to know which of the curated RefSeq transcripts you want to use is corresponding to which of the Ensembl transcripts. There are also evidence flags here, and I included a link to the website of the search of this gene. When you get these slides you can click on. The really nice thing for the evidence flags is they actually have hoverovers, and so it will tell you if there is a certain transcript level of evidence. So TSL1 is actually the highest level of evidence, and it will give you a little bit of information about that. And it will also tell you where it's been identified and the different flags for it. So for example, in this one, the 3' is incomplete in the coding sequence. And it's just really informative to mouse over those flags. The next transcript annotation effort that I want to tell you guys about is RefSeq. So this is the transcript annotation effort for NCBI. These transcripts are designated largely by curation, with RefSeq gene being the most thoroughly curated. These annotations are actually independent of the genome build. They are largely derived from GenBank, which is an effort that has collected sequences for about 260,000 species, and this is an NIH effort too. It was largely collected from individual labs, batch submissions, and large-scale sequencing projects. These are the prefixes you will most commonly see. So N is one of the most common ones. This means that the transcript is supported by some evidence. It might be published literature. It might be being seen in the GenBank database, as I had mentioned. X is actually a predicted transcript, and so we largely don't annotate or name variants on these transcripts. They're not necessarily confirmed with curation or evidence. And then the letter that you would see following N is either M, R, or P, M being for mRNA, R being for non-coding RNA, and P being for protein. So for example, the thing that you see most commonly is NM, which is an mRNA transcript that's supported by some sort of evidence, either a publication or a GenBank entry. And this is the predominant transcript set for clinical labs, clinical annotation pipelines, and for curators to know, most importantly, largely research publications, unless it's large data sets. So this would be relevant for gene curation. And I'm just going to take us out now to RefSeq. The website was just too large to do a screenshot of it, so. This is just an example of a gene in RefSeq. As I said, you would want to go to RefSeq gene because those are the most high quality curations. And you just have a little bit of information here. And actually the thing that you want to see the most, sometimes I'll take a look at the visualization of the transcripts just to make sure that the one I'm looking at is the one that I want for the number of exons and the length of it. If you hover over it, you can see the main features of it, the gene and the location and the coding sequence length and the protein length, just important things like that. You can download the sequence if you want and just click those. But the pieces of evidence that I like to see the most are further down. You can actually even look at RNA-seq data about expression across different tissues here, although I will show you GText for that in upcoming slides. I want to point out the bibliography. I've worked a lot with transcript curation for hearing loss with RefSeq, and our curations are included in their combination effort with Ensembl EBI LRG, which I'll talk about shortly. But there's a nice bibliography section for each gene. These aren't necessarily, I've been told by them, in a significant order. It's really just a random order of the number of genes, or of the number of citations. So if you're doing curation, you might just want to click See All Citations in PubMed and kind of skim through them. They might not all be 100% relevant. but these are papers that are given either by curators or by experts who have helped to curate the gene. So what you want to take a look at is actually the mRNA and proteins here. This actually gets pulled automatically into Alamut if anyone uses that viewer for their variant curation, but it will tell you the transcript and then a little description of it. So this is the longer transcript and encodes the longer isoform here. And then this one lacks an alternate in-frame exon compared to variant 1, just to give you a little bit of information about every single transcript when you're going through a transcript choice. And for example, this is non-coding RNA, so you would see X predictive, R non-coding RNA. Okay, let's go back. So now I want to talk about the collaboration between Ensembl EBI and NCBI, because I'm sure, as you're all familiar with doing variant and gene curation, it's definitely difficult to map across, and unless you have a useful resource like the table on the Ensembl website, it's kind of difficult to figure out what a variant is notated as in different transcripts across Ensembl and RefSeq. So both bodies, transcript annotation bodies, realize that this is a difficulty, and so they've really tried to collaborate. And their newest collaboration, which I'll talk about shortly, the main project, is actually a really helpful collaboration that I think is going to change the field quite soon. The first one that they started with is the Locus Reference Genomic, the LRG effort. So this aim was actually for clinical labs. It's to ensure efficient and consistent variant reporting by defining reference sequences. So this is actually usually a single transcript per gene unless you absolutely need multiple transcripts. This choice is a stable transcript, and it doesn't change. When I searched yesterday, there were currently 1,182 LRGs. So as you can see, this takes a lot of time because it's a lot of curation, and we really just don't have a ton of transcripts versus the number of genes at this point. So it's helpful if your gene has been curated, but if not, you're kind of out of luck in this case, which is why the main effort is valuable, which I'll talk about. So the LRG itself has curators, but it also solicits curation expertise from the community. And one of example of this, how I mentioned, is we actually collaborated through our hearing loss transcript curation with the LRG project. So after our transcript curation, which was published in the Journal of Molecular Diagnostics, I'll give you the citation later, we sent them a list of about 160 or so genes and their transcripts that we recommended, and they actually manually reviewed those suggested NM transcripts and updated them as necessary, and they tried to harmonize them with the Ensembl transcript to foster bidirectional exchange of data. And this curation effort will actually feed into the new main effort that I'll talk about. And this is the display here of an LRG gene that I had curated. And so it will show you the LRG, the length of it, the source for both the RefSeq and the Ensembl, so that's really helpful. And that will be listed here as harmonized transcripts, along with potentially a little bit more information about the gene. So the newest collaboration is the one that I'm most excited about, the main project. And this is the matched annotation from NCBI and Ensembl EBI project. And so it's actually a collaborative transcript set, which is the best of RefSeq and Ensembl, really, because it's going to be aligned to the genome build. A lot of the problems with RefSeq are that because they aren't aligned to the genome build, when the genome build and the transcript differ in a little bit of sequence, it can make alignment difficult. And this can be particularly difficult for clinical testing labs because a lot of times they align to the genome build, but then they will use RefSeq transcripts. So for example, we have a couple of genes in our clinical lab where we actually have to add or subtract nucleotide positions when we want to use a RefSeq transcript because it's not directly aligned to the genome build, and there is a difference between the genome build and the transcript. So in this case, it's going to be the best of both worlds, where it's the curation level of RefSeq aligned directly to the genome build. So this is actually going to be aligned to GRCH38, which is the newer genome build, and the hope is that this transcript set can be released right when gnomAD and population databases are switching over to GRCH38, and in hopes that most clinical labs would be able to switch their pipelines over to this newer genome build. And their goal is to have 100% identical sequencing for RefSeq and Ensembl, both UTRs and coding. And these transcripts are well supported, expressed, and conserved, and they're fairly stable, and the clinical and biological data is manually curated. And so there are two different levels of them, and the first level that they're working on is main select, which is 1 transcript per locus, and they actually were able to release 50% of these in December last year. Main Plus is kind of the upgraded version of that, and this is actually the evolution of the LRG project that I just talked about. They're well supported, they're tissue specific, and they're also relevant to certain user groups. So this will kind of be even a further level up from this main select. There are some really helpful resources. There was actually a webinar recently on this effort that was held. 50% of the main select transcripts, as I mentioned, are available now. So they're available on the NCBI's FDP site, which I included the link for when Jenny sends out these slides. And then they also have the webinar, like I said. They have listed the slides already here in this Google Drive. And according to Fiona and Joanella, a recording will hopefully be available soon. And it was a couple hours, and it was very, very informative. And they actually talked about the background of NCBI and Ensembl EBI and their transcript curation efforts. So I highly recommend that if you're interested in a little bit more background on these two annotation efforts. So I wanted to talk about how to map across transcript annotations because, as I said, a lot of publications will use RefSeq, clinical labs will use RefSeq, and a lot of resources will actually use Ensembl. So some resources with nomenclature will help you map. For example, the allele registry, and I'll show screenshots of these, the VCI using a VEP, and then also references themselves. Ensembl will do that, and if you're lucky and you have an LRG transcript, you can go to the LRG site like you saw in the screenshot, and it will tell you the RefSeq and the Ensembl, and it's essentially already curated for you. But here's a screenshot from the allele registry. If you're searching your variant of interest, it will list basically transcript alleles, and you will see the RefSeq transcripts and you'll see the Ensembl transcripts in case you need to translate across for different searches. Here's a screenshot from the VCI. It's the same thing. If you enter a variant and you search on the Basic Information tab, you'll see RefSeq transcripts and Ensembl transcripts so that you can see the HGBS for both. And then, as I showed from the screenshot previously, here's if you search Ensembl. This is a little bit helpful if you just generally want the corresponding transcript versus transcript. Say you're doing gene curation instead of just corresponding a variant annotation. You can just search it and it will tell you the ENST transcript versus NM. So the next thing that I want to talk to you about is where you can find information for curating transcripts. And this was kind of a trial and error thing when I curated the hearing loss transcripts that we worked on. So the transcript curation resources that I found most helpful, and I can say that unfortunately there really just isn't a lot of transcript information in the literature, and oftentimes it's difficult because even though RefSeq Genes does list all of the resources and all of the publications that are used for a RefSeq gene curation, it doesn't necessarily correspond the publication with the NM number. If you're lucky, the authors will mention or make it clear enough that you're able to correspond a certain RefSeq transcript with a certain publication, but sometimes it is tricky. So basically, the goal for transcript curation is try to figure out which transcript is the most relevant choice for you when evaluating variants, or if they're multiple, or if it should vary by disease for a gene. And so you want to figure out the expression profile and levels of each transcript expression levels to make an informed choice. And so things that I use are OMIM, a PubMed Google search to try and find primary literature. RefSeq Gene will also give you primary literature. GTex, which is really helpful because there often aren't a ton of publications in certain disease areas. And then also, I'm sure within different disease areas, there are disease-specific efforts, potentially RNA-seq datasets, or et cetera. So OMIM is really helpful in that there's a cloning and expression section. This is often the most useful. So as you can see for TBC1D24, there is mention of RT-PCR and the publication where it's found and the expression of the gene in multiple tissues. They might not necessarily list the transcript expression across multiple tissues in the OMIM entry, but it definitely helps to go and look in the primary pieces that they cite in hopes that there is specific information on different transcripts. The gene structure and the gene function sections can also provide relevant information, but I would always say go to the primary source because it will give you the primary images from gels on northerns or RT-PCR data that you can look at yourself and convince yourself whether you believe that expression or not. PubMed search feature isn't always super useful for me, but I do find that Google searches of your gene of interest in transcript expression, your gene in human transcript expression, your gene in isoforms, or your gene in splicing might help when you're trying to look for specific information about transcripts. And then GTex is actually one of my favorite resources because there aren't a ton of publications, like I said, on transcript-specific expression. This stands for the Genotype Tissue Expression Project. This is actually 53 non-disease tissue sites from about 1,000 adults. And these, keep in mind, are adults, and this is post-mortem tissue. That's something to keep in mind. So all tissues are not available for that reason. And they perform genome sequencing, exome sequencing, and RNA-seq. So we're interested in the RNA-seq. You can evaluate expression levels of the gene and transcript and exon level expression. So I'm going to take you there because that was another example of where it's just too much to show in a screenshot. So when you go to the home page, you just search for your gene. I will remind you that Ensembl transcripts are used for this. So if you have RefSeq transcripts that you're trying to coordinate with that, you should really go to the Ensembl website or the allele registry, some of the resources that I already listed to try to figure out which Ensembl transcript corresponds to your RefSeq transcript of interest. And so you'll see overall expression levels of your gene across different tissues, and TBC1D24 is associated with multiple epilepsy conditions, so you can see some higher expression in the brain. Unfortunately, we do have a little bit of an issue for hearing loss in that no inner ear structures, no cochlea is available in GTex, so we really just have to go with overall expression patterns, but hopefully you would get lucky and your favorite tissue or tissue of interest would be in here. My favorite use for GTex is actually the exon level expression. And so you'll go and see the predictive gene models, and you can see across each one based on color, the darker the color, the higher the expression, the specific exon level expression across each tissue. And so these would be the most expressed in the brain, which would make sense with our disease association, with the gray ones being, or light gray, or light blue ones being very lowly expressed. You can also look at splicing predictions with these junctions suggesting that this middle exon could be spliced out, particularly if it's in gray, it's largely not expressed. So these data sets predicting that this exon could be spliced out, and maybe if you had a loss of function variant in this exon, I'll talk about this more with my curation strategy, that it might not necessarily be a true loss of function because of this expression data in GTex. So GTex itself is just very helpful. I would suggest that if there aren't a ton of publications on your particular gene of interest. Okay, let's go back. So now I'm going to spend the last bit of time before I open it up for questions, talking about a specific transcript curation strategy that we did in hearing loss. So actually what we did is we took the 109 hearing loss genes on the otogenome test, which is the hearing loss clinical testing panel from the Partners Lab for Molecular Medicine, and we actually split them into 3 categories to kind of determine the level of curation that we should be doing on these genes. Category 1 genes actually only had 1 transcript, and so those would require little curation. Category 2 genes had multiple transcripts, but the longest transcript had all of the exons. So in this case, if you're a clinical lab, you would most likely pick the longest transcript. This might not be the most biologically relevant transcript, but these hopefully would require a little bit less transcript curation than category 3, which was multiple transcripts were present and each transcript might have unique exons. And so we were actually surprised to find out that across all of these 109 hearing loss genes, they're actually pretty evenly split across all categories, really pointing out that this tricky category 3 really requires transcript curation, and so just genes in general require transcript curation. And I would assume that this is not unique to hearing loss and that this might hold true across different disease areas, but I'd be interested to do some more curation across different disease areas and try and find out. And so what we did was manual curation at the gene level and the variant spectrum, which I'll talk about. And these are actually really important because it would impact the classification of variants, largely the loss of function classification. And this would be corresponding to the ACMG code PVS1, which is very strong evidence of pathogenicity. So you essentially only need one more piece of evidence of pathogenicity to classify a variant as likely pathogenic if you classify it using PVS1, which is loss of function variant and loss in a gene where loss of function is a known mechanism of disease. So to put this in perspective, for category 1 genes, if you have a loss of function variant, the variant is likely loss of function if the mRNA doesn't escape NMD or if the variant isn't present at the very end of the gene, something like that. In category 2 and in category 3, if you have a loss of function variant in a gene with multiple transcripts, you really need to do more evaluation and more curation on these because you need to ask yourself, is the transcript biologically relevant and is the exon biologically relevant? Maybe it's predicted to be spliced out in GTEx. Maybe the transcript isn't expressed in your tissue of interest that you're evaluating. So these are things that would need to be further curated before you can just apply PVS1 and almost bump a variant to likely pathogenic. If you had, for example, absent from population databases, PM2, that would bump it. And these are the categorization results. As I said, there was a pretty even split across all of the categories. Here are all the transcripts for all of the categories. Obviously, category 3 had the most transcript, and the number of exons for all of those categories. And so for category 2 and category 3 genes, which were 71 genes, we wanted to do further curation of these genes, really to evaluate what would be the consequence of loss of function variants in these genes. And so what we did was we looked at the gene level data and the exon level data for these genes. So for gene level data, we wanted to look at transcript expression. Where was the transcript expressed? Is there information in the literature? If there was, we were very lucky and we used that. If not, we went to GTEx and tried to look across all tissues because, like I said, we don't have any expression data for cochlea. And we used this to classify exons. So for exon level variation, we actually wanted to look at high frequency loss of function variants in gnomAD, suggesting that if there's a high frequency of a loss of function in a particular exon, maybe that exon isn't necessarily included in the relevant transcript, especially if loss of function is a known mechanism of disease, and that would maybe make an exon or a transcript suspicious. And so we actually used this to classify the exons. And once exons were classified, we used this to evaluate interpreted variation. So I'll start by giving you the classification definitions of the exons and then explain what this would potentially mean for interpreted variation. So we designated the exons as either clinically significant, exons of uncertain significance, clinically insignificant, or non-coding. So non-coding is self-explanatory. Clinically significant exons, we tried to be very cautious with this. We wouldn't want to rule out an exon, particularly for variant classification in a clinical lab, unless we were really sure that the exon was not significant. We don't want to miss anything for patients. And so for a clinically significant exon, we wanted to see that there was evidence that the exon is required for biological function. Was there evidence in the literature? When we looked at GTEx, was the exon posed to be expressed at high levels across multiple tissues? For exons of uncertain significance, these were quite common. There is no expression data in the literature. Potentially, they were predicted to be alternatively spliced by GTEx, like I showed you in the one example. And they had one high-frequency loss of function variant in gnomAD. And we defined high frequency as greater than 0.3%. This is actually our general likely benign cutoff for hearing loss in Lab for Molecular Medicine, and I think now the hearing loss BSEP. The clinically insignificant exons were confirmed in the literature that they weren't expressed in our tissue of interest or clinical relevance, and or they had more than one high-quality, high-frequency loss of function variant in gnomAD, so greater than 0.3%. And so once we classified all of these exons, we wanted to look at interpreted variation across these exons. So we actually pulled variants from ClinVar that had at least one star, so assertion criteria provided, or DM variants from HGMD. We know these aren't super reliable, but we just wanted to have a measure of interpreted variation in these different exons that we had classified. And so we pulled pathogenic and likely pathogenic variants from ClinVar and looked to see if they were present in exons that, based on our curation, might have questionable significance. And this is just the classification here. There were 24 to 36 non-coding exons. Each one of the categories of genes actually had exons of uncertain significance. And even though there weren't a ton of them, because we tried to be cautious, there were actually 8 clinically insignificant exons that we came across, largely in category 3, which makes sense.
Starting next week for math and the following week for staff. And the schedule will
I think there's some background noise if people want to mute if they're
no one's here
not
today
talking.
to help out, but like I said, next week if you're looking for math.
Can everyone mute,
and the
please?
third week if you're looking for staff.
It was not really a math question. It was more like
okay, I'll just continue on. So there were 7 clinically insignificant exons in Category 3 and 1 in Category 1. And there were a handful of clinically of uncertain exons across all of the categories. But there were a large number of clinically significant exons because, like I said, we tried to be cautious in ruling out exons.
In lieu of having the high school.
So I want to give you an example of this exon level classification that we did, just to make it more concrete for you. And so I'm using the gene endothelin 3, which is associated with Waardenburg syndrome. It's pigmentary abnormalities and potentially hearing loss syndrome. It's autosomal recessive. So RefSeq lists 5 transcripts, which you can see here. All of these share exons 1 to 3 here. Light blue is non-coding and blue is coding. And also 5. However, exon 4 is not present in all of the transcripts, and that's boxed in red. There's actually a frame shift variant in this exon in 0.6% of Finnish European alleles in gnomAD, including 2 homozygotes. So in theory, those individuals would have Waardenburg syndrome, which is a pretty rare syndrome. And when we looked in GTEx, it predicted that this exon is actually spliced out where endothelin 3 is expressed. So when we looked at classified variation in this exon, there is actually 2 DM variants in HGMD that are located in this exon. And it really calls into question the pathogenicity of those 2 variants because we ended up classifying this exon as an exon of uncertain significance. And so what I mean by the pathogenicity of the 2 loss of function variants is largely just that you can't look and say, this is a loss of function variant. It's causing loss of function. And in this case, it is the second to last exon, so you might not feel that way. But if it was, for example, in this exon, you can't just say that it's causing loss of function. I'm going to apply PBS1 and call it likely pathogenic. We really have to evaluate it a little bit more. Is it observed in cases? Are there publications about it? Just examine it a little further, short of just saying loss of function, likely pathogenic. And so we wanted to look at the impact of interpretation, looking at these interpreted variants in these exons of uncertain significance and clinically insignificant exons. And so 6% of all exons were actually exons of uncertain significance across these genes. These exons contained 124 clinically significant variants, so either likely path or path ClinVar variants or DM variants in HGMD. And as I mentioned in the previous example, these variants would really require further evaluation to determine if other data support pathogenicity, because looking at the exon landscape, we really can't find significant data implicating the exon being important in the transcript in the gene. And something I want to end with is really just a mention of telling curators that they should try and think about technically challenging regions. So it's not a curator's job to evaluate the technical aspects of testing. However, there are some genes that have very well-known sequencing issues. And it might be important to be aware of these when scoring variants for gene curation and also just classifying variants in general. I can say that experts are perfect for this, so even if they aren't 100% experts on the ClinGen gene curation framework, they should be able to tell you, oh yes, this gene has high homology with a pseudogene. You really need to find other ways to detect variants for this. You can't use NGS. And this could even be part of the pre-curation. We did this for a few hearing loss genes. Just like choosing a transcript choice could be part of a pre-curation for gene curation. If it's a common problem, very well known, publications might allude to it. One of the gene curations we had for hearing loss, they actually alluded to the pseudogene issue, or actually it was an issue in the reference sequence in the genome build that it was inverted incorrectly. So we had to look and make sure that all variants that were present in the sequence were actually Sanger sequenced. And so we evaluated this in hearing loss, and to just give you one example, we took NGS data from our lab and then from the Children's Hospital of Philadelphia, and we actually found 43 technically challenging exons in 20 different genes. And these had inadequate coverage or homology issues that might lead to false variant calls. So you wouldn't have to go this far, but it might be important to just check while or before you're curating a gene if there might be technical issues that might cause incorrect scoring for variants in certain areas of the gene. And one example that's very well known in hearing loss is stereosilin, where there's high homology to a pseudogene. And so when you look at the mapping quality or the depth of coverage, it's actually the first half or so of the gene that has high homology to the pseudogene. And you can really see that looking at the combined NGS data here. So like I said, you don't have to go as far as we did for this project, but something to keep in mind when you're curating. And so future directions for this transcript curation effort that I did, scaling it is a challenge. It was very manual, and it would be great to do this manual of a curation effort across different disease areas, but you might be able to do some auto-categorization of category 1, 2, or 3 genes using RefSeq gene annotations, and then also maybe some automatic filtering using loss of function frequency filters for gnomAD, and also GTEx expression. But limitations being it's a manual process, and also GTEx can only help you so much because it contains adult post-mortem tissue. Maybe relevant tissues and time points might be missing if you're looking at a neonatal disease or if you're looking at cochlea in our case. So in conclusion, I hope I've convinced you that transcript curation is critical for variant and gene curation, and that annotation efforts are harmonizing, which we are thrilled about. It's going to make curation a lot easier for us, and that there really are resources available to curate transcripts, even though the literature isn't super descriptive about every single transcript for a particular gene. And with that, I'd like to acknowledge the people who helped me with the Journal of Molecular Diagnostics publication. And that is all I have, so I can take questions.
That was fantastic. Thank you so much, Marina. Does anyone have questions?
What time would that be?
It's not loading for me.
Excuse
Hi,
me,
this is
can
8:45. Okay. And how long does it
sorry,
take?
can you put yourself on mute if you're not talking on the conference call, please?
Kathleen Wallace at UN. First off, yeah, um,
Hi, Kathleen. We
personally,
can hear you. Go ahead.
sorry, yeah, there's some background noise, so I think it's a little bit.
Let me see if I can see who that is and mute.
Have a direct question, more along the lines of I think that this information would be hugely useful in kind of like a guideline or new curation educational materials. I'm guessing that that's probably in the works already, but I just wanted to suggest that maybe even going a step more
Yes.
basic.
Well,
Yeah.
let's see. This is the 22nd. And there's not. That's the first available or earliest appointment.
Oh, okay. Sorry, I think that's.
Sorry, I'm going to mute everybody and then unmute you, Kathleen. Okay.
Okay, thank you.
Let me do that. I'm so sorry.
It's okay. I thought. Anybody hear me yet? Oh,
Okay.
there we go.
Yep. Yes, I can hear you
Awesome.
now. Yep.
Perfect. Okay, so my comment would be that if this is getting worked into providing like training materials or education materials, I think it would be really helpful to get even to more basic levels of information. So I know as a junior curator and coming in with limited background, very limited background in clinical genetics, there were it was a steep learning curve trying to understand just all the acronyms and where these different transcript like nomenclatures came from, and like I think it'd be immensely helpful to have kind of like a learning document that teaches information like this in one place, because I just haven't been able to find. A lot of my learning was a lot of separate Google searches trying to figure out, okay, what does this acronym mean? How does this relate to theirs? So things like that, just I think would be very helpful moving forward, especially with things like community curation and the different levels that we're looking into. So thank you very much again, Marina.
Of course. Yeah, thanks for that comment, Kathleen. I totally agree with you. When I started this transcript curation effort for this paper, Heidi had to tell me that RefSeq was not aligned to the genome build, and Ensembl was directly aligned to the genome build, just because it's so difficult to find in their FAQs any of this simple information like that. So I agree. I think we should maybe put some of this in some training materials, because it's been a painful and steep learning curve even for me to try and find all of this. And I think with the main effort, they really have tried a lot more education and to just be more transparent in their processes. But yeah, I agree. And we should do something like that.
Yeah, this is Jenny, and I think that's a great idea too, and thanks so much for raising that, Kathleen. So that's definitely something that we can discuss as part of the Biocurator working group, because part of our function is, of course, to generate some educational materials. So, we can definitely think of that with Marina's help, of course. So, yeah, that's a really great idea. Thanks. So I just unmuted everyone again. Does anyone else have any questions for Marina?
Marina, this is Christy at UNC. Would you mind showing us the GTAC website, like where you got the information on the Wardenberg gene, just to like walk us through it, or walk me through it?
Sure. So first of all, like I said, it's a little tricky because you have to map across your RefSeq transcript of interest to your Ensembl transcript, because these are Ensembl transcripts, the one that I have pulled up. So I likely recommend going to the Ensembl website, finding your RefSeq transcript of interest, and which one it corresponds to. So you know which one of these that you should be looking at and which one of your exons. If you don't have a way to map your RefSeq to your Ensembl, you can also use genomic coordinates, which I'm mousing over here if you can see it. And so as a quick and dirty way to do that, I just looked at the genomic coordinates to find which part of the transcript for Ensembl that your exon of interest would fall in. And then you want to go up here and look at the specific exon level expression. It kind of gives you a key here of the darker being the higher expressed in different tissues, and it lists all the tissues here. And so the most highly expressed exons would actually be very dark ones here. So these end exons, and the exons expressed in the cerebellum there, whereas very lowly expressed ones are light blue or even gray. And so in this case, exon 2 is largely in gray, and you can see this little loop here, suggesting that there's alternative splicing and that exon 2 is largely spliced out in all of these tissues because of its low level expression. However, exon 1 is also kind of lowly expressed too. But is that kind of what you're looking for, Christy?
Yeah. I wasn't quite confident in predicting what would be spliced out based on the information you showed earlier, so that was